today is day one, and
In this video, I'll share my experience genetically modified E. coli bacteria so it could grow in conditions it normally wouldn't be able to. And that's thanks to CRISPR. Alright, in order for any organism to grow, it needs to make proteins using its DNA code. And all the proteins are made by the ribosome. The media we're going to grow our bacteria on has an antibiotic called streptomycin that prevents the ribosome from making proteins, meaning that our bacteria can't survive and grow. That's why we'll make a mutation in the ribosomal subunit protein, RPSL, that generates resistance to streptomycin, allowing our E. coli bacteria to grow and enjoy its life. Okay, now let me show you a step-by-step process. So there are three main parts of this experiment. The first one is growing and preparing the bacteria. The second is modifying, and the third is incubating and hoping that our CRISPR works. And we're starting with making plates.
To dissolve the agar, we put it in the microwave for a few seconds at a time so the bottle is not boiling over, just placing the lid on top. It will take 2 to 4 minutes for the agar to be clear with no visible particles. Flipping the plates upside down, store them in the fridge at 4 degrees. Now it's time to work with E. coli bacteria, adding sterile water to the bacteria tube using our pipette. Spread the bacteria on the LB agar plate we just made using an inoculation loop. Wait until the agar is dry, put a lid and flip the plates to store. Let them grow at 37 degrees for 1 to 2 days. That's how I did it, using a homemade incubator. And we should get something like this. Hey, it's our bacteria. Wait, but how will we insert foreign DNA? Cell walls will prevent it. But if we insert specific components, CRISPR will work. That's why we'll add the bacterial transformation mix that consists of PGF-8 and calcium chloride to our bacteria. Gently scrape the E. coli off of the fresh plate using a thin side of the inoculation loop. Mix the bacteria with the transformation mix. Make sure any big clumps disappear.
Before we continue, let's talk about CRISPR, which is an immune system in bacteria.
During a viral infection, CRISPR array is transcribed into a long RNA. Then this RNA is broken into smaller pieces, called CRISPR RNAs, each containing sequences that match viruses' RNA, so basically memory of past infection. This process is mostly done by trans-activating CRISPR RNA. Together, they bind to the protein known as Cas9. This complex then scans along DNA looking for a matching sequence, and if there is, Cas9 will make a cut in this DNA.
In plant and animal cells, these breaks are replaced by small changes or new pieces of DNA. Since we're talking about bacteria, it will die if we don't introduce a DNA template. And that's our chance to insert modified donor DNA.
Here is our Cas9 guide RNA and the DNA template. And then we need to add 55 microliters of sterile water to each component. Now, add 10 microliters of each DNA tube, Cas9 guide RNA in DNA template to the bacteria we just made. Remember to change out the pipette tip for a new one. Force DNA droplets to the bottom of the tube. Now it's time for heat shocking. Put the tube in the fridge for 30 minutes, then incubate for 30 seconds at 42 degrees. For all bacteria to restore, take the LB media microcentrifuge tube and add room temperature water to it. Then add this LB media to the cell mixture with our DNA. Incubate at 30 degrees for 1 to 4 hours, but it's better to do for 12 hours to increase the chances. While CRISPR is doing its work in the bacteria, it's time to prepare the agar media with the antibiotic, streptomycin. Just repeat the process of making plates, but now use the LB strep media. Using an inoculation loop, gently spread the bacteria around the plate. Let it dry for 10 to 15 minutes. Put the lid on top and flip the plate upside down. Incubate for 24 to 66 hours at 30 degrees.
Alright, as we can see, I got completely different results, and that's really exciting. I decided to continue my experiment and see what would happen in a few days.
If you had an idea that was going to outrage society, would you keep it to yourself? Thanks for watching.